హలో 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 రాట్లే గారు వస్తుందా ఇక్కడ? దీంట్లోకి రాట్లే కదా. వస్తుంది వస్తుంది ఇప్పుడు వస్తుంది. వ్యూ వచ్చేసి ఫాలో హోస్ట్ వీడియో హైడ్ సెల్. హలో రాట్లే గారు వస్తుందా ఇక్కడ? దీంట్లోకి రాట్లే కదా. వస్తుంది వస్తుంది ఇప్పుడు వస్తుంది వస్తుంది చూడు. వీడియో బ్లాంక్ వస్తుందా? ఫాలో హోస్ట్ వీడియో హైడ్ సెల్. హలో ఇదేంది వస్తుందా ఇక్కడ? దీంట్లోకి రాట్లే కదా. వస్తుంది వస్తుంది ఇదే కదా వస్తుంది చూడు. వీడియో బ్లాంక్ వస్తుందా? ఫాలో హోస్ట్ వీడియో హైడ్ సెల్. హలో ఇదేంది? అది కాదు, ఒక నిమిషం సారీ అది కాదు, వేరే షేర్ చేస్తా. ఇదే కదా ఇది షేర్ చేయాలి. వీడియో బ్లాంక్ వస్తుందా? ఫాలో హోస్ట్ వీడియో హైడ్ సెల్. హలో ఇదేంది? అది కాదు, ఒక నిమిషం సారీ అది కాదు, వేరే షేర్ చేస్తా. ఇదే కదా ఇది షేర్ చేయాలి. వీడియో బ్లాంక్ వస్తుందా? ఫాలో హోస్ట్ వీడియో హైడ్ సెల్. హలో ఇదేంది? అది కాదు, ఒక నిమిషం సారీ అది కాదు, వేరే షేర్ చేస్తా. ఇదే కదా ఇది షేర్ చేయాలి. వీడియో బ్లాంక్ వస్తుందా? ఫాలో హోస్ట్ వీడియో హైడ్ సెల్. హలో ఇదేంది? అది కాదు, ఒక నిమిషం సారీ అది కాదు, వేరే షేర్ చేస్తా. ఇదే కదా ఇది షేర్ చేయాలి. యూట్యూబ్ డబల్ వస్తుంది, మీకు వస్తుంది సౌండ్ చూడండి కావాలంటే. వస్తుందా? ఓపెన్ చేయండి. సున్నందుకు చేసిన అమ్మాయి కనపడుతుంది కదా అక్కడ. వస్తుంది అమ్మాయి కనపడుతుంది చూడు. ఆ అమ్మాయి కనపడుతుంది ఇక్కడ దీంట్లో. బెస్ట్ ఓకేనా సార్? ఇంకా మీరు జూమ్ ఓపెన్ చేసుకొని మీరు వర్క్ చేసుకోండి. ఇది అదే అది మన ఇది ఇప్పుడు అది యూట్యూబ్ లింక్ పంపిస్తున్నాం కదా. పెట్టింది కదా.
'Cause I'm lost in the way you move, the way you feel.
क्या दिख रहा है?
A medical college in a private university is the first medical college under
on innovative approaches in drug discovery and AI technologies, which is organized by School of Pharmacy, Anurag University, and sponsored by MedHub Student Community and IVY Overseas Consultancies. And a heartfelt welcome to our chief guest, Dr. Kala Kumar, sir. Thank you, sir, for joining us today. And also our registrar, Dr. Balaji, sir, registrar of Anurag University. Thank you, sir, for joining us. And FDP, the Faculty Development Program, is designed to accelerate the recent advances in pharmaceutical sciences and healthcare through artificial intelligence and innovative technologies. Our FDP is indebted to provide a comprehensive and dynamic platform for professors, researchers, and industry representatives to get aware on latest AI technologies and methodologies. So now let's start our program by inviting our delegates onto the dais. So I would like to welcome our Dr. Vasudha Bakshi ma'am, Dean, School of Pharmacy, to come onto the stage. And Dr. Balaji sir, Registrar of Anurag University. And Dr. Kala Kumar sir, our chief guest, General Secretary, Indian Pharmaceutical Society. And Dr. Krishna Prasad sir, Associate Professor, School of Pharmacy, Anurag University. Light is a symbol of wisdom and prosperity. It is believed to bring welfare and positivity around us. So to light up the lamp, it's an honor to welcome Dr. Vasudha Bakshi ma'am and Dr. Balaji sir and Dr. Kala Kumar sir and Dr. Krishna Prasad sir to please light up the lamp. And also I would like to welcome Prasanna from BPharm third year to sing a prayer song.
गणनायकाय गणदैवताय गणाध्यक्षाय धीमहि गुणशरीराय गुणमण्डिताय गुणेशानाय धीमहि गुणातीताय गुणाधीशाय गुणप्रविष्टाय धीमहि एकदन्ताय वक्रतुण्डाय गौरीतनयाय धीमहि गजेशानाय भालचन्द्राय श्रीगणेशाय धीमहि एकदन्ताय वक्रतुण्डाय गौरीतनयाय धीमहि गजेशानाय भालचन्द्राय श्रीगणेशाय धीमहि.
Now I request Dr. Vasudha Bakshi ma'am to please address the gathering.
A very good morning to our respected registrar, Dr. Balaji Utla of Anurag University, chief guest of today's FDP program, Dr. Kala Kumar sir, professor of veterinary sciences. He's also a general secretary of IPS, co-conveners, Dr. Krishna Prasad, associate professor, and Dr. Sunan, assistant professor, faculty members, and delegates. So I welcome you all for this 1-week FDP program on innovations in drug discovery, AI technologies, which we will be conducting for the next 5 days in a hybrid mode. So as a part of Anurag University teaching and learning processes, we conduct such a number of FDPs, which will enhance the knowledge of the faculty in the recent trends, because AI is being extensively used for number of drug discovery program. Like if you see the drug discovery program, the usage of this AI is on a very high theme. And the theme of the program basically focuses on drug discovery and its advancement, usage in the AI technologies, and advancement of informatics. So the CAD use and this AI usage is basically enhancing for the drug discovery program for a number of drug molecules. Initially, we used to follow the traditional methods for this particular drug discovery program, where then too the hits used to come very less. But with this extensively modified use of this AI, which has number of software, if you see recently the usage of this sort software in the AI, it is coming in a lot. In the CAD also it is coming. There are lots of opportunities. So it is one way of drug discovery. For it is helping both the faculty as well as the students to explore this part. New startups are coming with this AI technologies, where there is a combination of both the CAD as well as the drug AI molecules. So this use of this computational methods has created a pathway in understanding and screening the potential lead candidates for any. If you see the number of diseases which are coming at a molecular level is also increasing at a very high. So within this short time, if you see a traditional drug discovery program, which is of 12 years, costing such a billions, with this AI, the number of cost involvement is also reduced. The time which is used in the drug discovery is also reduced, because we are going to utilize all the different in vitro techniques. So from the life-saving drugs, drug discovery, development and production, to clinical trials and drug target identification, artificial intelligence, if you see, it has made a lot of advancements, not only in CAD. If you see in case of pharmaceutics also, we are traditionally we are using AI for GI stimulation techniques. We used to use various models. Nowadays, this AI has been utilized in pharmaceutics also. We are using not only in pharmacology, but we are using in all the different specialized agents. This AI has been extensively being used. So if you see, the pharma companies have also started using these techniques and adopting more efficient and computer assisted processes that incorporate expert decision as well as quantitative analysis, because as we are in the discovering mode, like we are discovering new therapeutic identities. So that is why it's a combination of both, which is being extensively used in the AI. So the next evolution of this approach, artificial intelligence to advanced studies, now it is also being utilized in bioinformatics extensively, because of these high-end technologies. We are having number of drug approval, the cost effective therapy. If you see the cost of the drugs in the cancer specifically, if you quote the cost of the drug, how it is, the cost is very high. Or if you must have heard, like chemotherapeutic drugs, they cost very high. One cycle of that 15,000, 14,000. So by using such extensive techniques of such AI, we can try to reduce the cost of the drug, because the time involved in the production of that particular drug, it is more. That is why the cost adds up. So if the cost, if just 1 mg of the drug of cancer drugs, if you see, the cost is very high. Why is the high? Because the process of synthesis, the process of purification of the drug, it is a very extensive, and the yield of the drug, whenever you manufacture any component, the yield also very important. So these are taking very much time. So that by using this AI technologies, you can reduce this. So today's FDP, like we have devised this 1 week FDP, we have number of resource persons who will be letting you all the different softwares, different methods which we'll be utilizing. And today I thank Kalakumar sir for accepting an invitation to be a resource person for this FDP. And also today we have Dr. Tyagi, another principal scientist of CCCAC, and Dr. Muhammad Eris, who is the senior principal scientist of CCMB. He will be highlighting of different in vitro models like zebrafish models and everything. How do you utilize them for conducting this various experimental studies instead of using the animals for your particular pharmacological studies? So they'll be enlightening us and expertising of how to use this artificial intelligence approach. So with this few words, words. I welcome all the resource persons and also for accepting an invitation. Also, I thank our registrar, Balaji sir. Like whenever we call, he's definitely like being available and supporting us for all these various events which we are conducting in pharmacy. So I thank sir. And also, I thank my sincere appreciation to co-conveners, Dr. Krishna Prasad and Dr. Sunan, for carrying out all these FDP in a very short time. And I thank all the team of pharmacology for assisting them. So I would like to thank the sponsors who have sponsored our event. So during the next 6 days, you'll be learning the different topics, which are the very new different things which you can utilize the final year students in their project work. It will definitely help them to give a good publication. So I hope that you will understand all these concepts in a more productive way to make a more smart decisions in your further project program. Well, I don't want to take much of your time. So keep learning, keep updating yourself with the recent trends. With these few words, I thank you one and all. Thank you.
Thank you, ma'am. Now, I would like to welcome Dr. Balaji sir, Registrar, Anurag University, to deliver his words.
Our chief guest of today, Professor B. Kalakumar Garu, General Secretary, Indian Pharmacological Society, Professor Vasudha Bakshi, Dean, School of Pharmacy, and Dr. Krishna Prasad, co-convener of this faculty development program. My dear professors, and I do see there are many students here. The reason why I agree to programs that Dr. Bakshi plans is because I worked in public health. I mentioned last time. I was involved in designing 108 services, and I was the founding CEO of 104 services, telemedicine, dial a doctor, and every month we used to send vehicles to the villages to provide healthcare to people. So health is one of the important things in a person's life. Many of you have seen in COVID, people have been pauperized. People who had 1 acre, 2 acres, small homes, sold it to save their loved ones, parents, children. I know one family, middle-class family, their life savings, they had some 25 lakhs or 30 lakhs. 32 lakhs was the bill for the lady when she had COVID in one of the private hospitals. So healthcare is very expensive. One of the key things that we talk about in healthcare is equity. By equity, we talk of 3 A's: accessibility, availability, and affordability. Services and products could be available, but not accessible. Accessible and available, but not affordable. Affordable, but not available. Any one of the A misses, we believe there's no equity in healthcare. And pharmacology, pharmacy, innovation, discovering, reverse engineering, all of these things are very expensive processes. Thanks to India, thanks to companies like Cipla, thanks to many companies in India, they make drugs so accessible. Africa is thankful to India. Otherwise, when the epidemic of HIV/AIDS was going on, some countries' population between the age of 15 to 50 got wiped out, as if somebody took a brush and painted white, and people are gone. So a country cannot run. There are only elderly people and very young children. So societies get destabilized if there is not proper healthcare. We have seen what has happened in COVID, the pandemic that we have seen. We hope and wish we don't get another pandemic, but there is a need for us to keep developing new drugs as people develop resistance to drugs, and improper use of drugs leads to many drug resistance, especially the TB in India, which is prevalent in many places, especially poor people get affected. Almost every human being in India, they say, carry the TB in some form or the other. And there's a multi-drug resistance right now. So there is always an imperative for us to keep developing. not only for diseases like cancer, simple diseases like malaria, where poor people die. We haven't, only recently they developed a vaccine for malaria. I think government of India approved for one vaccine. Why did it take so many years to develop a small thing? Go to any tribal area. Go to just 200 kilometers, Srisaram, you go, Chenchus. The population is dwindling. You go to Adilabad, the Gonds, population is dwindling. Go to Bhadrachalam, you have Koyas. These are all malaria endemic areas. So I think one of the key things is to use knowledge to make the world a little better place to live, especially for those people who cannot fight for themselves, who are struggling to live and survive. The society itself is iniquitous in many ways. While some of us are billions of dollars, $1 billion is about 8,500 crores, billions of dollars. Some people don't even get 100 rupees a day. But the dignity of survival, existence, healthily is an important right of human beings. So health, I think, should be a right, and education is the other one. So in that, Dr. Krishna Prasad and Professor Vasudha have thought about a faculty development program. I'm a big votary for faculty development. The reason being, knowledge is getting obsolete very quickly. The shelf life of knowledge is coming down, just like idli flour or ID batter you get, idli batter you get, basi ho jati hai. Har ek 3 saal mein, knowledge is getting thrown out. What you learned 3 years ago is sometimes may not be relevant. At the same time, it's so exciting because new tools are coming. Earlier, drug discovery used to take years and tons of money. It would anywhere cost $1 billion to $3 billion. That means anywhere between 10,000 crores to almost 30,000 crores to discover one molecule or develop something. So if we can reduce it, crash the time it takes. I'm told Moderna, for which they got a Nobel Prize, there's also criticism as to why they got it. Leaving that controversy apart, they could develop that protein in just less than 6 months. And thanks to many of these discoveries, such a pandemic could be contained. Though millions of people died all over the world, with heart-rending scenes in a developed country like Italy, struggling for beds, and elderly people who died, the children couldn't see them off. They were just sent to cremation directly. We have seen what tragedy disease can bring. So pharmacy is something that you alleviate the suffering, misery, disease of people. So the faculty must develop themselves constantly. What we call CME, we call in healthcare, continuous medical education or continuous development. Without that, we don't go anywhere further. Since this is a hybrid kind of a session, I would invite all the participants to be active in the sessions. Virtually, sometimes what happens is when we are attending something, our focus goes here and there. Please pay attention, because they have invited some of the best minds, some of the best professionals, like Professor Kala Kumar and other people. Use that efficiently. Ask questions. One way to keep yourself awake in a virtual meeting is to ask questions. Take notes, ask questions. There is no right or wrong question. All questions are legitimate. So please do ask questions, because the whole knowledge movement happens by asking questions and challenging. You know how science moves forward. For those other people in Hyderabad, other pharmacy colleges where faculty are there, I would invite them to work with us closely. Since we are a university, we have AIML department, we have computer science department, we have electronics department, and so many of them are there. If you would like to participate in collaborative research, because research is something that no single person can ever say that I have discovered everything. Newton once said, if I can see further, it is because I stand on the shoulders of giants. So the people who before us who did so much of work, that is what knowledge we are standing on. So, knowledge constantly keeps changing. We keep invalidating things, validating things. Karl Popper said, science is about conjectures and refutations. That's the philosophy of science. You make a conjecture, you falsify it. You make another conjecture, falsified. That is how, in Telugu we say, munduku mooredu venakku baredu. 2 feet forward, 2 feet backwards, 4 feet backwards. Science moves like that, but that will be frustrating. I appreciate the scientists. They never lose whatever they choose to do for their lives. Many of them don't get celebrated. Many of them don't get celebrated. So there's a Sanskrit shloka. I'll share that and stop after that. Prarabhyate nakalu vigna bhayena nichaihi. The lowest of the people are scared and afraid to start something because they believe there'll be obstacles. Prarabhya vigna nihata viramanti madhyaha. The mediocre people start something with a lot of bang. And once they find some obstacles, they abandon the task. Vignair muhur muhur api pratihanyamanaha. Even if you're encountered again and again and again by obstacles, prarabhya, after starting, uttama gunaha, great people, na parityajanti, they don't abandon. I wish you all a wonderful learning sessions for the next 5 days. Thank you very much for inviting me.
Thank you, sir. Now it's time to invite our chief guest, Dr. Kala Kumar, sir. And so just a minute. So we have a short profile of Dr. Kala Kumar, sir. He has done his graduation in 1985 and post-graduation in pharmacology and toxicology in 1988 and PhD in pharmacology and toxicology from SVVU in 2010. And currently he's the professor and university head and Department of Pharmacology and Toxicology in CBSE Hyderabad. He received the State Best Teacher Award in the year 2020 and was awarded SB Pandey Oration in 2017 and Dr. Lalita Kameshwaran Oration in 2018. And he has executed projects funded by ICAR, state and industry sponsored projects. It has 68 papers in national and international journals. He has conducted many training programs and also he's a resource person for various universities and central government institutions. He's a member of committee for scrutiny and selection of faculty. So let's invite Dr. Kala Kumar for his valuable words.
So, very good morning to all of you. Dignitaries on the dais, the Registrar of Anurag University, Dr. Balaji Garu, the Dean, Pharmacy Stream, Dr. Vasudha Garu, Dr. Krishna Prasad and Dr. Sunan, the coordinators of this faculty development program. Thank you for inviting me for this program. Maybe one reason could be that as I am the current General Secretary of Indian Pharmaceutical Society, so the organizers might have thought that it would be appropriate to involve the society. So because by virtue of my position, I could be here as a chief guest. But before that, when I think 5 or 10 years back, when Dr. Diwan was here as he was the dean or the director, I don't remember. But when he was here, I was a regular visitor as an examiner as well as a resource person. So once again, it makes me nostalgic. I was expecting that there would be faculty, but I find that there are future faculty over here. Good, that's nice. That you're going to develop yourself to become faculty. And Dr. Krishna Prasad was mentioning that there are PG students also, PG students in pharmacology. That's good. And pharmacology is a dynamic subject. Both my earlier speakers, that is Dr. Vasudha Garu and Dr. Balaji Garu, they have emphasized the incorporation of the technology into pharmacology. If something is happening, pharmacologists would be the first persons to grab it, because until and unless we are open to the happenings around us, we get obsolete, means we get outdated. To prevent that, we need to update ourselves. AI and ML, artificial intelligence and machine learning, is a new tool, but we need to incorporate this tool so that we can come out with the best that we can offer to the society. So with this intention, this particular faculty development program has been chalked out, and I have selected my topic as gut microbiome and neurodevelopment. That is my area of interest is neurodevelopment, CNS and neurodevelopment. And in that, I really enjoy cognition, that which encompasses learning and memory. So when we learn well, it gets fixed in our mind. I think you know about dendrites, right? And on the dendrites, you have the spinous process. If I ask you to recite the second or the third table, you will do it very fast. If I ask about 23, you may fumble, because you have recited it n number of times. That's the reason why you say that 2 into 10 is equal to 20. That has become a long, that has been transformed into long memory instead of short memory. Short memory is that if I ask you what you ate yesterday, midday or night, then if it is something like chicken or mutton, you would immediately say that this is what I ate. If I ask the same question regarding the diet that you took on Tuesday midday or Tuesday evening, the last Tuesday midday or the last Tuesday evening, you might not, because it's not etched in your memory. Whereas something that we learn and try to repeat it n number of times gets into long-term memory. That's what I'm going to talk about. But before that, I thank the organizers for inviting me to this artificial intelligence and machine learning. But more than that, the word faculty development program has attracted me. That made me spare some time to come and speak to the faculty. No doubt, even if the number of participants is less, let us not get worried, but because everyone in the faculty, in the teaching faculty, they may not be interested. Those who are interested, they will be in a position to develop themselves. So the number does not matter at any cost or at any point of time. Number does not matter. Even if the participants are less, it's good. It's good, because those who are serious about it, they only participate. So if the faculty, if you allow me, I just want to mention that I worked in the government department for 13 years. And in my 40th year, I jumped into the university for the single point that I wanted to be a catalyst. If I can teach my students well, and I would be in their memory, and whatever the subject I teach, that would be in their memory for the next 40 years. I am from veterinary profession. So that made me shift. Of course, in the process, I lost my seniority in the university. I lost my financial benefit also, but still I was passionate. In one word, I say that teaching is a fungal infection. Those who have fungal infection, they come to the faculty development program. That's my, if you ask me, that's my idea. Yeah. Yeah. That's the reason why we make you sit for hours together. Even if you are not interested, we just hammer you so that you take the subject, right? So with these few words, thank you very much.
Thank you so much, sir. So now I would like to welcome Dr. Krishna Prasad sir to deliver the oath of thanks for today.
A graceful and warm morning to all, each and everyone, and welcome to all delegates. So before going to vote of thanks, it's my privilege to share my hearty thanks to each and everyone, those who are participated in this FDP program. So this is it is the right time to what at the right end of the session. So the inauguration session. So we are going to discuss about that all those things. So here we are conducting the 9 important sessions. So that is going to be within 5 days. So it is very useful. And I thankful to our Dr. Kala Kumar sir. And in spite of his busy schedule, he accepted our invitation. And one of the most thing I want to share with. So I called sir just one last 2 days back. So sir has given that immediate permission to associate with the Indian Pharmacological Society. So for that, I'm very thankful to Dr. Kala Kumar sir. And it's very deep, my deep sense of gratitude to our Registrar Anurag University, Dr. Balaji sir. So sir also has given permission to come over here and to give that valuable presidential keynote speech, and also is being here motivating that all activities of in Anurag University. And and also my sincere thanks to our Dean Vasudha Bakshi ma'am. So as a convener of this FDP program. So without her support and guidance, the FDP program is not possible. So thank you so much, ma'am. And also I thankful to my all colleagues, and especially my hearty vote of thanks to all my pharmacology department. So those are all done good organization of this first day of FDP program. And I also thank to that all participation, those who are participated in this FDP program through online as well as offline mode. So please be alert and concentrate on this all 9 sessions. So before going to that conclusion, I would like to share one small inspirational words to those who are participated in this FDP program. Okay. So first of all, I will say that here 5 I's is there. I letter. So this is the FDP program for innovative approaches in drug discovery. So before going to that innovative approaches in drug discovery, first we have to have that here deep discovery ourselves, those who are participated. So if deep discovery, how it will become based on that 5 I's. So I will tell that here first I. So this is the good platform to provide that all sessions like information. So everyone will get that information through various sources, social media. This is the first one, I. So second I also is there. So that is the interaction with that all session. Those who are high delegates and high mentorship also is there. So interact with that all sessions so that you will get that a lot of information. This is the second I. And third I is, so we have to implement. So information finished. So interaction, as Dr. Balaji sir said that here questions are very, very important. So quality of questions will give that here a lot of quality of information. By the interaction only it is the possible. So third I, that is the implementation. So as usual, all here, we across that around 130, above 130 delegates are presented here. So how many of you implemented, I don't know. So please, whatever you are learning this whole 5 days, 9 sessions. So these are all very valuable sessions. So you have to implement in your research area. And last one, after implementing, you can consistently implement. So that is the integrate in your subconscious mind. That is the I is integrate into your subconscious mind. And last I is, so it will go deep inside in your unconscious mind. So whatever you are learning that research. So these are all the 5 is very, very important. Information, interaction, and implement, that should be very consistent. So it will go directly into the subconscious mind, that is called as inside, integrate. Then last, finally, inside. So if anybody will do this all 5s, then you will say simply, I did. I achieved in my research area. So this is what I want to share with you all, those who are participated. And thank you so much for giving this good opportunity as a welcome to all, each and everyone. Thank you so much. Thank you.
Thank you so much, sir. And thank you, ma'am and sir, for sparing your time on stage. So I request you all to please join the audience.
Thank you, sir. Thank you. Oh, sir.
I request all the faculty members to please join the stage for a small group photo.
Able to open my PowerPoint. Yeah. So how many of you are PGs? PG in pharmacology? Yeah, right, good. Good. Sutix? Sutix? None from Sutix? That's it. Right. How many of you are from WhatsApp University? WhatsApp University? None? That's good. Right. So, yeah, right. So can I have the pointer? Pointer? ఇది వచ్చింది అచ్చా 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 ఓహో రైట్ యా సో ఈ మైక్ కదా So, hope I would be in a position to catch your attention. That's a challenge for me today, early in this morning, right? My talk is on microbiome, gut brain axis, right? And actually, I thought of speaking about neurodegenerative diseases, but I think I'll make it a little bit. I'll just try to modify a little so that it would be interesting to other students and other faculty from other streams also, right? So I want to impress upon this particular aspect, that is, you are male and female because of the hormones. If someone is said to be a male, because of the hormones, male hormones, he is a male. If someone is female, because of the female hormones, she is female. Good, right? If hormones can differentiate the sex, are responsible for the sex differentiation, right? Neurotransmitters are responsible for your behavior, right? Yeah, are you able to catch this point? Neurotransmitters are squarely responsible for your behavior. And now, if you're not able to sit in your seat properly, and if you wriggle like a caterpillar, right, then blame your neurotransmitters. Even if your faculty comes to you and says, "Oh, why are you doing like this?" Say, "Sir, neurotransmitter, it's not me," right? Sir, you'll have to blame my neurotransmitter, right? And ultimately, the synthesis of neurotransmitters is based on what you eat. What you eat, right? So, first of all, for your behavior, you want to blame the neurotransmitter. And then you can also blame the diet, right? Whatever you have, whatever is your diet, you can just blame that, right? So the entire presentation would be that, right? And this is an upcoming area. Microbiome is an upcoming area. Hitherto, we did not have so much of research focused on this. But now the entire research is being focused into this particular area. Of course, it's only one part. I don't say that the entire research has taken a U-turn and it is into the microbiome. No, it's a part. It's a part. It's a part because ultimately it is responsible for cognition. For cognition. You may ask me as to what cognition is, right? Now you have seen me occupying the chair on the dais along with the dean and the registrar, right? And the organizing secretary, right? And now if you go out and in the evening someone says Kalakumar, right? You say that you don't know because you have seen me, you had the first picture, you have taken a first picture of myself, and then now when the name comes to your mind, you try to recollect. That is recognition. So the first picture is cognition, the second picture is recognition. That's all. It's only you're trying to retrieve. In the first instance, you learned, and the second instance, you're trying only to recollect whatever you have learned. If your learning is proper, then your collection would be easy. Then the process of recollection would be very much easy. Able to understand this one, right? So learning and memory, both of them, they form cognition. So I want to impress today that we, based on our diet, our microbiome means the microbial population in the colon. We say that if I ask one of you, you will say that I am from so-and-so colony, some Brindavan colony, some Nagarjuna colony, Krishna Nagar colony, some colony, right? So also the microbial organisms, they colonize in our colon, means they also have a colony, right? And their address is our colon. In the large intestine, you have the three parts, right? So now, that's where the microbes they harbor, they live, and they colonize. Hence, it is called colon, right? Yeah, colony, area where we live, right? So also the microbes, they stay there, right? And it's a case of symbiosis, mutual benefit. If I am there, I should be of some benefit to you. And if you are there, you should be of some benefit to me, right? You are of a benefit because at least I can look at you. Otherwise, if it were to be empty chairs, what is the fun in looking at the empty chairs, right? And you have the benefit that I talk something so that at least your time is passed, right? So this is what is symbiosis, mutual benefit. So also the bacteria, they have the mutual benefit, right? So in turn, they come up with the secondary metabolites. They utilize the undigested food that you, the undigested part of the food that you have taken or that you have ate, right? And they survive on that. They survive on the undigested food that which we take, and in turn they produce secondary metabolites, primary metabolites that are responsible for our health and well-being. And cognition is a part of the normal well-being, right? And I said neurodevelopment diseases. I also, neurodegenerative diseases. So I also have taken up neurodevelopmental diseases. I think you might have heard about autism. In case of autism, social interaction would not be there, right? They would not be in a position to properly communicate. And also they have this repetitive behavior. You might have come across many articles in the media. Of course, now print media is going down, right? And you have social media. But my advice in the classroom and wherever I'm invited as a resource person is that less of Facebook, more of textbook. Not even Google. Google may just try to clarify, but it will not give you the full detail, or it will not give you the full explanation, right? So less of Facebook, more of textbook. Textbook will certainly help. At least go through the diagrams, go through the figures and the tables. That would be sufficient. That would be really sufficient, because today textbooks are into they are coming out with more figures. Even journals also. Journals, they're asking for figures. They're asking for illustrative diagrams, so that at one glance I should be in a position to understand. I should be in a position to understand as to what that particular aspect is all about, right? If at all there is a 10-page article, if it can be abridged into a very small figure, I think that would be more beneficial for me. That would be time saving for me. And if I'm interested, I'll get into the deeper part of it, or otherwise I'll just flip it, right? Able to follow that? Now, with that, we'll go to the next slide. I need to show this way, or I need to show this way? Yeah. So we have what is known as Barker's hypothesis. Barker's hypothesis, that is, you are what you are you are what you were in the womb. Means it has long been decided as to what you would be today, right? And the influence of the environment is minimal. The influence of your genes is maximal, right? So provided you are exposed to a particular environment, your destiny has already been decided when you were in your mother's womb. That is what is Barker's hypothesis, right? And today the saying is that you are what you eat. You are what you eat. So we need to be careful about what we eat, right? Yeah. Because ultimately the diet is responsible for the maintenance of the brain, for the neurotransmission. And if neurotransmission is not proper, behavior is improper, right? That's the reason why in the classroom, you would be looking at WhatsApp, right? If some friend has sent a message, but unfortunately, you are so insignificant that you don't receive any message, right? And then the frustration is writ large on your face, and then you're taken away from the classroom atmosphere. But by chance, if there is a message, you would be trying to you'd be trying to give a reply, but you would be nodding your head. That's the beauty of sitting on the other side of the table, right? Yeah, right. So it's రావడం. So shall I say next and ఇది రావడంలే. Yeah. ఇది రావడం లేదు. I'll just say next. So coming to the microbiome, microbiome is nothing but colonization of microorganisms. That is the microbiota. Right? Yeah. And symbiosis, I told you. And then dysbiosis is something opposite, right? And I think in high school we are familiar with this word, attention standardies. Standardies means, be at comfort, right? Be comfortable, right? Stand at ease, right? It is not stand at ease. It is not STANDATIS. It is stand at ease, means be comfortable, right? And when we say disease, it is discomfort. Right? Disease is nothing but discomfort. That's the reason why we say pathology is study of diseases. Study of discomfort is pathology. Right? So we have dysbiosis, that is rise in pathogenic bacteria, and you have neurodegenerative diseases, that is Alzheimer's disease, Parkinson's disease, multiple sclerosis, amyotrophic lateral sclerosis, and then you have Creutzfeldt-Jacobs disease, that is CD. And then you have neurodevelopmental diseases, autism spectrum disorder, and attention deficit hyperactivity disorder. As faculty, as some of your faculty, you might have experienced this. The fellow is less attentive, but more hyperactive, right? And it takes your energy, it takes your intelligence to maintain this particular fellow, right? And if wrongly handled, or if you don't handle, if you try to ignore him, he'll spoil the entire class. He'll spoil the entire class because he would be the pacemaker, right? And he would be the pacemaker. And we know that the two tissues that are excitable in the entire body, one is the cardiac tissue and the other one is the nervous tissue, right? So he would try to transfer his energy to the other fellow, so that the other fellow would also be excited, and then there would be chaos, and 50% of the class would not be listening to you, right? And you have neuropsychiatric diseases, anxiety and depression, and reversible neurological diseases like migraine, POCD, that is post-operative cognitive dysfunction, and long COVID. Early diagnosis and treatment through conventional and novel treatments, right? They would be restoring this microbiome so that the person would be normal and he would be healthy. Next. Right. So this is the definition of cognition. Cognition is the mental action or process of acquiring knowledge and understanding, right? And encompasses all aspects of intellectual function. Intellectual function, language learning. Some of you can learn the language very easily, right? Even if you go to Tamil Nadu, in 6 months, you can learn Tamil well, right? And even if you go to Karnataka, you can learn Kannada well. But others, all through their life, they cannot learn it, right? They may learn 2 or 3 words, right? But not more than that. And this is one of the cognitive function, right? Under the dominance of the cerebral cortex, right? So cerebral cortex is responsible for the cognitive functions like language learning, mathematical ability, that is computational ability, reasoning, logical reasoning. Some of them, they're quite logical, that they apply that logic and then they come out with the statement. Logical reasoning, numerical ability, I told you. So all of these, they come under cognition. So cognition also encompasses learning and memory. That's what I told you. If you want to be a good learner, then your cognitive function has to be good. So I request you to eat well, eat properly. So I'll show you what is the best diet. What is the best diet? so that our cognition and learning capacities are good, right? And how best we can treat the neurological disorder. The primary neurological disorder is that inability to learn and memorize. And as faculty, we express this difficulty whenever there is viva voce. I think now the viva voce is there by external examiners, right? Yeah, then that's good. That's good. No, that has to be there. That has to be there. It's not that otherwise it would be too homogeneous. It has to be, the examination process has to be heterogeneous. Someone from outside, the very thought that he is not our own, but from outside, that would create that fear psychosis, right? And on the day of examination, you would not eat properly. Even if it is puri also, you hate puri that day, right? You dislike puri for that particular day. The reason is that because of the anxiety, right? And this causes a change in the microbiome, a change in the gut microbiome, right? And why actually, see, examination, what it has to do with the gut? It has to do with the brain. But gut is responding, isn't it? Gut is not comfortable. Till the examination is over, the gut is not comfortable, right? Yeah. And if at all, if you don't fare well in the examination, and then there would be lacrimation, right? And lacrimation is also under the influence of the CNS, right? Okay. Yeah. So what has the gut to do with the brain? Yes, there is interaction with brain and gut. That's what we are going to discuss. Next. So so neurotransmitters, as I told you, they are responsible. Can I have the pointer? Can I have the pointer? Can I have the pointer? Oh, he took it there. No problem. So do you have a long stick? Hey, children, do you have a pointer? You have this keychain, no? Or otherwise, if you want to attract someone or to divert someone, you would be playing with that pointer keychain. You don't have? First of all, you go to the doctor and please check whether you're boys or not. You don't have a keychain with the pointer. Are yaar, kya hai yaar? You know, at least you have a cigarette lighter. Cigarette lighter? No. How about this side? Cigarette lighter? No. Oh, outdated. You're not keeping in tune with the times. Okay. Right. So neurotransmitters, they are responsible for emotion, anxiety, mood, behavior. Just now we took the example of an examination, and now through the vagus nerve, it supplies to the visceral organs, and it's not only afferent, it's also efferent, AE, right? It not only brings signals from the brain to the visceral organ, that is the gut, but it also takes signals from the gut to the brain, right? And then the gut synthesizes neurotransmitters and is also responsible for the neuroendocrine system, that is neuroendocrine system, neuroimmune system, and also the hormones. So ultimately, brain, it controls gut motility, enzyme secretion, microbial balance, nociception. Nausea is pain. Pain perception is nociception, and it is affected by stress and behavior. Next. Yeah. So brain and body health. Yeah. If at all, if brain is affected, the cognitive impairment would be seen. Means the learning and memory would be lost, right? And then you have the behavioral changes and altered neurotransmission. That's what I'm talking about, that you are what you are based on your neurotransmitters. If you are in the classroom and if you're behaving well, your neurotransmitter system is good. Otherwise, it needs to be altered or it needs to be treated, right? Because it's a neuropsychiatric disorder to be in the classroom to look at WhatsApp, right? Gut, healthy microbiota, immune tolerance, intact gut barrier, microbiota. And on the unhealthy side, you have microbiota dysbiosis, which is responsible for altered gut function, If the gut function is altered, the brain function would be altered. That's simple. My presentation is over. I'm just impressing upon you that based on your gut activity, your brain activity would be there, right? If gut activity is proper, brain activity is also proper, right? Okay, the liver can regenerate, but nervous system cannot regenerate. That's the reason why you have Alzheimer's disease. Of course, we all suffer with Alzheimer's disease in the examination hall, right? The question would be there, and you do whatever you want. You look at the teacher, you look at the wall, you look at the roof, nothing would come out because you have studied, but yet you have forgotten. Alzheimer's, amnesia, loss of memory, right? Yeah. Next. Yeah, the same thing, the neural network, the neuroendocrine system. I told you about the neuroendocrine system, isn't it? That is hypothalamic, pituitary, adrenal axis. And we know that adrenal gland is responsible for the production of cortisol, all right? So ultimately, if there is stress, and then if it is acting on the gut, then that is due to this, the cortisol, the release of cortisol through this system, that is HB axis system. And gut immune system, if the gut immune system is good, if the gut bacteria are good, then there would be less production of inflammatory cytokines. Otherwise, there would be increased production of inflammatory cytokines, which would affect the brain, right? And this inflammation of the gut would cause a leaky blood-brain barrier, and then it is going to cause neuroinflammation. Neuroinflammation would be the first sign for the neurodegenerative diseases, right? Inflammation followed by actually injury somewhere, inflammation and injury somewhere, right? Injury to the gut, it might have resulted in watery motion for you or the loose stools for you. Hey, either of you neurotransmitter, but they are not functioning well. Come here and sit. Come. Of course, it's part of the classroom behavior, I know, but still, you caught my attention. Come. అది వెనక చూస్తా ఉన్నాడు పాపం. నేను కాదనుకోని వెనక చూస్తా. Come. No, the next. Yeah, no, the next one to you. Oi, yeah, come. Be a part of the faculty. Come over here. Right. Good. And in most of the faculty interviews, before you are taken as a faculty, one of the questions would be, how would you control your classroom? Right? Of course, we are all faculty. We have faced that. See, controlling the classroom is equivalent to presenting the subject. Both of them go hand in hand. Okay. Yeah. Right. So gut microbiota are responsible for the production of neurotransmitters. And if the gut health is affected, then the barriers would also be affected. That is the intestinal mucosal barrier at the first instance, then the blood-brain barrier would be affected, and it would affect the brain, right? And so we had neuropsychiatric diseases, reversible neurological diseases, the neurodevelopmental disorders, and also neurodegenerative diseases. Neurodegenerative diseases, already degeneration has set in. That's the reason why all that we can do is only improve the quality of life, but you cannot cure an neurodegenerative disease. But you can improve, the existing condition. Instead of worsening, you can improve. But Alzheimer's disease is one of the worst disease in the sense that you can't differentiate between food and water. My own aunt suffered, and she was in a position and she was not able to differentiate between food and water. And she used to give a vague look at her daughter. And even when her husband would come, my uncle would come, she would give a vague look. It's really, really bad, right? I think the pharmacology, as pharmacologists and from the stream of pharmacy, we need to come out with a drug for all these neurodegenerative diseases. At least, if not, See, if we are not able to cure, at least we should be in a position to stop further damage, right? Even if that is done, I think that would be helpful to mankind, right? Next, that is the gut-brain axis. And coming to the GAT microbiome, bacteria, yeast, virus, and archaea, that is extremophils. Extremophils, excess pH, they can survive. Excess temperature, they can survive. Excess salt, they can survive, right? So you have archaea also, right? Y colon is that GAT movement is slow, and the undigested nutrients, they serve as food for the bacteria. And we were saying that appendix is a vestigial organ. Now, due to this microbiome research, scientists have come out with an explanation saying that if appendix is removed, we are removing a place for the bacteria, right? And if bacteria are not there, then the gut-brain axis would be affected, right? So appendix is no more appendix. Generally, what we do, if you read a book, at the end you have appendix or appendices, right? Appendices is plural, right? Appendices means something that is added to this one. So we were saying that on the right side you have this appendix, right? And people would suffer with appendicitis also, right? So we used to say that it's a vestigial organ. Now it's no more a vestigial organ. It's no more a vestigial organ, and it has its own importance, right? Now, so these are the bacteria: Clostridium bifido, Lactobacillus, Ruminococcus, and Akkermansia. I think you are familiar with 1, 2, 3, and this one. This one is the latest. Akkermansia is the latest, and it is emerging. Means the next time when you go to the doctor, he may prescribe yes, a species from Akkermansia genus, right? And role in our metabolism, these gut bacteria, they produce neurotransmitters, hormones, metabolites, and other bioactive molecules, which maintains intestinal barrier, blood-brain barrier, and regulation of immune system. Next. So how do we identify? Because the fecal matter, it contains so many bacteria, but amongst all, how can we differentiate and say that this belongs to this genera, this belongs to this species, and this belongs to this phylum or the family? How do we do this? We do this by fecal culture and microscopy, the most conventional, whereas next generation sequencing, the next step, that is next level, here we go for 16 years rRNA to identify the genus level. But if you want to identify to the level of the species, then you need to go for whole genome sequencing, where the DNA would be identified, and that would be that would help you to identify the species. And then you have metatranscriptomics, metaproteomics, and metabolomics. In metatranscriptomics, you identify the mRNA, which is responsible for the production of the proteins, right? And metaproteomics, analysis of the protein; metabolomics, analysis of the metabolites. Based on this, based on this, right? So based on this, you can identify which bacteria is present. Why? In certain of the diseases, in certain of the neurodevelopmental diseases, in certain of the neurodegenerative diseases, some of the bacteria, some of the good bacteria, they would be lacking. So in such cases, we need to give bacteria as a drug. Actually, bacteria is called as a drug, is called as a bug, right? That's why when we say, when we use microbial resistance, we have this word. Are mere venaka baga tondara chestunaru, mundadu goosobedta. I'll make you faculty. Right. Okay. Please, tondara cheyakandi. Okay. Just take 1 or 2 points from the entire 30 minutes of the lecture. I don't say that no one will have that capacity to take the entire 30 minutes lecture, but we can take 1 or 2 points. Okay. It would be good to hope you are not from MBA. right? There is none from arts and the arts and MBA and all that, no? Everyone is from pharmacy. మరి చచ్చినట్టు తినాలి, ఏం తలకాయ తిరుగుతుందా? Oh no, I thought that they could be from other faculties. So okay, you can leave. But if it's pharmacy and you don't want to listen the latest developments, no, nothing doing. Hey, Babu, vastaledamaa. Right. Yeah, this is the next slide. Right. Okay. Right. So coming to the microbiome development, it begins after birth and stabilizes at 3 years, right? And delivery method, natural versus cesarean, would also affect your microbiome, right? And microbiome today, it is being worked upon, and it is being emphasized that ultimately one day, based on your fecal culture, I can certainly tell what you are.
Yeah.
Just 1 gram of your fecal matter, right, will give your entire picture as to what you are and why you are. Means your disease pattern, your disease profile, your behavioral profile, right, and what you are likely to be in the near future, right? Yeah. Right. So gestational age, preterm or full term. At times, if someone is troublesome, we'll say that fellow is premature, no? Right? So premature births, they do have their own disadvantages, right? And feeding, breast versus bottle, solid food introduction, annaprasana, right? Transition to cow's milk, diet in teen and adult years, right? Actually, I had the temptation of writing Swiggy and Zomato over here, but I didn't, right? And then you have the hormones and the environment, right? And one thing is that microbiome is specific for each individual, including twins. Even if you have a twin, no, nothing is going to work out. You have a different microbiome from your twin, right? So it's something like, I think one day Aadhaar may be replaced by your microbiome. It's so specific, right? Yeah. And geographical influence, yes, if you emigrate to US, your microbiome would change because your diet has changed, the dietary pattern has undergone a change. Next, sir. Yeah. Now, able to see that here you have a different color in this pie diagram. You have this represents the bacterial family, but still these two women, they are twins, and still they have a different bacterial culture. And next, and at one point I read a T-shirt. I have this habit of reading everything, even name boards and all that. So I read that. It said, who said I do not have, who said I'm cultureless? I have sufficient bacteria, right? It's oral culture. Means he was talking about the bacterial culture in his mouth, right? So his question was, who said I'm cultureless? I have sufficient, right? Yeah. Okay. Good bacteria, they produce what are known as short chain fatty acids. And these short chain fatty acids are anti-inflammatory in nature, and they are responsible for the intestinal health, right? If at all the intestine is leaky, and then from the intestine, bacteria and other unwanted substances, they gain entry into the systemic circulation, they cross the blood-brain barrier, and then they are going to affect the brain, right? CNS, right? And when we call them as bad bacteria, when gram-negative bacteria produce, when the bacteria are of gram-negative nature, and they are responsible for the production of lipopolysaccharide, which is responsible for inflammation. So then we call it as bad, right? And the next slide. Yeah, you're able to see this one. I think this single slide would emphasize our topic, that is diet low in animal fat and protein, but high in plant fibers. Plant fibers, prebiotics, their nature is that they are prebiotics, right? Yeah. So host enzyme digestion, acid hydrolysis, and then you have microbiota related to metabolic health, right? So these are the microbes that have gained prominence or that have developed based on your diet. And here you are able to see that this is a different diet, high in animal fat and protein, but low in plant fibers. And because of the proteinaceous nature of this diet, you find that the proteolytic bacteria, they have taken upper hand, and it results in the form of in the production of peptides. And these peptides are broken down into sulfur-containing amino acids, other amino acids, and aromatic amino acids. Children, I want to give a specific point over here. Many a time, there would be production of gas, either due to carbohydrate or protein diet, right? Carbohydrate diet is orderless, whereas protein diet is full of order because of this proteolytic bacteria. Proteolytic bacteria, they gain upper hand, and you have these are the amino acids that are released, and and also aromatic amino acids like tyrosine, phenylalanine, and tryptophan, they are released. And here you have a foul smell, whereas here in carbohydrate diet or a plant-based fiber diet, you don't have any smell, right? The next slide. So in continuation of the first the above slide, you have short chain fatty acids, which are called and there would be increased mucus secretion, decreased luminal pH, and decreased methane. And we are not talking about the stomach, but we are talking about the intestine over here, right? So you have propionate, acetate, and butyrate. To be precise, not even the intestine, we are talking about the colon, right? We are talking about microbiome, the presence of the bacteria in the human body. They are in the colon, and this is what they do. They produce short chain fatty acids, provided your diet is made up of plant material, right? Yeah. So you are ultimately these propionate, acetate, and butyrate, they are going to act on G protein coupled receptor 41/43, and then they are going to produce these gut hormones PYY or GLP-1, that is glucagon-like peptide 1, and then this is going to decrease the blood glucose, increase energy expenditure, and also when you come to the other side, that is animal protein based diet or just pure protein with less amount of plant material or the fibers, then that decreases the mucus secretion and increases acetaldehyde, increases the luminal pH, and then is responsible for increased blood glucose, metabolic endotoxemia, and decreased glucose stimulated insulin secretion. The next. So this is what is going to happen, right? And these are the, in case of obesity, these are the bacteria that are going to whose production is going to be affected. And these are these two and these three, totally 5, they are going to increase, right? And here also, in case of malnutrition, I have projected this slide that to make you understand that these are the dysbiosis that is going to be seen in case of obesity, malnutrition, type 2 diabetes, and next. Yeah. And in case of metabolic liver disease and cardiometabolic disease. So all the microbiome is related to the production or genesis of almost all the diseases that are all the diseases of the vital organs, that is the liver, the heart, the brain, everything is going to be affected. One thing is that I did not come across, after going through the literature and all that, I did not come across the word lung, right? Somehow lung is lung seems to be out of the process, right? But people might not have done much research, because even lung is also exposed to the outside environment. We are talking about gut because it is exposed to the outside environment, directly like a skin, gut, right? Yeah. Next. Right. So coming to the diets, man in his early days, he was a hunter-gatherer. So the diet is called hunter-gatherer diet. Now it is grain diet. From rice, it's slowly going towards millets. Ragi dosa, special ragi dosa, and we order that. Right? If someone says that, what is special in your hotel? He says ragi dosa, then we are interested in that. Right? But ultimately, it's all grain diet. That's what I wanted to impress upon you. Rice to millets, and then Hyderabad is said to be the biryani capital. Right? You have the Pakistan team. Even in Pakistan also, biryani is famous. But after coming here, these people, they were not going to the net practice, but they were searching for biryani outlets. Right? Yeah. Western diet, energy dense, sugars, fats, proteins, low intake of fruits and vegetables. And this is one of the areas where dysbiosis results. And mind you, in one of the seminars, I learned that it is good that we eat pani puri at least once in a month. Of course, right? At times we may fall sick by eating pani puri because he dips his hand and there could be fecal coliforms. But still, after we are not satisfied with 5 or 6 pani puris, we ask for one more. See, one would be there in the mouth, and then we'll say, auric, right? Right. But E. coli, pathogenic E. coli, because ours is a poor country, we don't maintain hygienic standards, right? We are susceptible for this E. coli, but E. coli releases one of its metabolite, which is anti-cancerous. That's the reason why West Indies and people from the Western developed countries, they suffer with colon cancer, whereas we do not suffer much when compared to them, right? Thanks for the pani puri, right? So I advise you to eat pani puri at least once in a month, right? And make sure that it is not a working, the next day is not a working day, let it be a holiday. Right. And then you have the Mediterranean diet, full of legumes, fish and nuts. And Mediterranean diet is said to be the best diet. The best diet. As on today, Mediterranean diet, they live long. Right? And it is full of legumes, fish and nuts. And and this one gives you a balanced microbiome, good short chain fatty acids, and it prevents obesity, hypertension, cardiovascular diseases, and Alzheimer's disease. Vegetarian or vegan diet is also good because it contains good amount of plant fibers. Whole grain diets, grain diet is different and whole grain diet is different. Whole grain, where the polishing is less and it's not refined. High carbohydrate and fiber is seen with these whole grain diets, and you also have FODMAP diet, which is beneficial in diarrheal IBS. The other day, I went to the doctor. I have been diagnosed with diabetes in the month of April. So I went to the doctor and I said that these days I'm not comfortable with my stomach. And then he said it could be IBS. And he made me wait. And he was speaking to another patient, and he said today IBS is quite common. All right, irritable bowel syndrome. Okay, right? Yeah. And slowly in youngsters, that's what this is my observation. In youngsters, inflammatory bowel disease is on the rise. It could be due to TB. India is endemic for TB. It could be due to TB, or it could be due to Crohn's disease. There is a disease called CROHN apostrophe S, Crohn's disease. All right. So inflammatory, and if someone is diagnosed with IBD, now these physicians, they want to differentiate whether it is TB or IBD. Crohn's disease, right? Yeah. The next slide. Yeah. Coming to the individual foods, Coffee, tea, and berries. So it's classroom scenario, 59 participants and you people. It's like a classroom. Individual foods, coffee, tea, and berries. So they increase Ocelobacter species, which is anti-inflammatory. Red wine fish, they favor the microbiome. High protein, fat, sugar, they cause dysbiosis. But ketogenic diet, somehow it has been found out that ketogenic diet, it reduces seizures, obesity, insulin resistance, and improves metabolic health. If metabolic health is good, mental health is also good, right? It goes without saying, right? And exercise, yes, you need to exercise. You need to exercise. Yeah. Of course, you may say that Puneet Rajkumar and others, they have this six-pack body, but I have a family pack, right? Not me, you youngsters, most of you have family pack at this age, right? The reason that they might have succumbed because there could be there could be a congenital anomaly, right? Right from their birth, there could be congenital anomaly, or they might have developed the formation of thrombus in their teenage years, right? So that's one of the reasons why teenagers are suffering sudden heart attack, right? Sudden cardiac death, right? Oh, no, next, go back to the previous one, right? So controlling your emotions, fear, anger, sadness, this also alters your microbiome, right? And if we are on the road, we can't control our emotions, right? So if I want to shout at someone, I just keep my car door shut, that I raise my windows, switch on the AC, and then I shout so that the other fellow does not listen. But I have given vent to my anger. So that's lost. It's not there in my heart, so that it does not produce any sadness. Next, sir. Yeah. So these are the factors: diet, antibiotics and medications, aging, early childhood experience, host genetics. Next, sir. Cause of dysbiosis, diet changes, aging, that is, as you age, your gut changes from anti to pro-inflammatory, right? And lifestyle, urban, dust and humidity, fog. So please don't select Delhi as your next destination. Be away from fog, because fog is dust and humidity is going to disfavor your microbiota, right? And rural, yes, soil favors microbiome, right? And we do not know why it happens, but those who are living in the rural area, they have a very good microbiome, right? And environmental, heavy metals, pesticides, smoking, toxins, infections, and diseases, they are going to cause dysbiosis. Socioeconomic factors, alcohol, food diet, sedentary lifestyle, and lack of hygiene. Drugs, proton pump inhibitors, selective serotonin reuptake inhibitors, and side effects of drugs, other drugs, they are going to cause dysbiosis. Next. Yeah. So this is one representative figure where you have the afferent vagal nerve fibers, efferent vagal nerve fibers. Of course, I told you, probiotic foods, probiotic beverages, and symbiotic supplements. They all help in balancing or restoring the microbiota. So once gut health is proper, then the gut-brain axis, right, would be proper. And if the CNS is proper, then the mental health and the neurodevelopment would be at its best, right? Otherwise, what happens is that if the microbiota is improper, then it could be due to antibiotic therapy or whatever it is. The microbiota of the microbiome is imbalanced or disrupted, result in gut dysbiosis, and there would be cognitive weakening. Cognitive weakening is that your learning would go down, your memory would go down, and you would be suffering with lack of intelligence, right? Yeah. Next. The same thing, of course. So here, if you look at this is the healthy microbiome, and on to the right you have a dysbiotic picture. Here, the bacteria, they are responsible for the production of short chain fatty acids. In turn,
it stimulates the immune system, and it also keeps the gut epithelium intact. And if the gut epithelium is intact, there is no entry of outside microorganisms, that is those which are present in the lumen, into the systemic circulation, right? And even you have this enteric nervous system. Enteric nervous system, we only, when we talk about the peripheral nervous system, we talk about the autonomic nervous system. Autonomic nervous system consists of parasympathetic nervous system and sympathetic nervous system. But now, maybe by the time you graduate and then you become teachers like us, enteric nervous system is going to be a major nervous system. All right. Slowly, a chapter is going to be added to the existing textbooks, wherein the study of enteric nervous system would be a must, right? And the role of enteric nervous system. That's the reason why, if you see something something that is not pleasant, then you feel a change in the stomach, isn't it? All right. Actually, of course, if it's too strong for you, and if you cannot withstand it, you may feel dizziness or reeling of the head, and then you may fall down. But if you are able to withstand it, what would happen is that you will feel that your stomach is upset, right? Yeah. So this is nothing but the enteric nervous system. Enteric nervous system. You have seen with your eyes. The stomach has not seen, or the gut has not seen. It does not have any eyes, but still the message has gone, has percolated to the gut, so that now you find that there is a change in the gut. The gut motility is affected. All right. And on that day, you skip your lunch. Right? You might have observed it a number of times. You skip your lunch, or you're disinterested. You're completely disinterested. You have lost your appetite because of the emotional situation that you have faced. Right? So this proves that enteric nervous system plays a major role. Oh, no. Get back. Right. So one more connection is through the vagus nerve, right? Now coming to the disorder, reduced diversity or reduced microbiome, leaky gut, lipopolysaccharides, they are absorbed and they enter the brain, and slowly they start the injury, inflammation, and then the disease. So the disorder would be behavior, neuroinflammation, neurodegenerative disease, and neurodevelopmental disease, right? Now, if at all, if we are careful to maintain the homeostasis, there would be proper appetite, cognition, behavior, and neuroprotection. If especially we take youngsters, parents, they take their youngsters to the doctor and say that, sir, we tinnadanle, sir. And then he'll say, are you worried? Are you worried about something? What worry has to do with his appetite? Yeah, there is absolute connection with worry and appetite, right? Because there is the gut microbiome is affected. That's the reason why he is not able to feed. The next, right? So next, we'll skip this. Yeah, we'll skip this one also. Right. So these are this is what I have been saying, that short chain fatty acids. These are the metabolites contributed by the gut microbiome, and these short chain fatty acids are responsible for so many functions. And one of the important one is butyrate. Butyrate is anti-inflammatory in nature, and butyrate would try to prevent the inflammation of the intestine. If the intestine is healthy, then the brain is also healthy. Next. So even vitamins are also produced. See, unlike in case of human beings, we veterinarians, we never prescribe a vitamin for ruminants, that is cow, buffalo, sheep, and goat. We don't prescribe vitamin preparations, no vitamin injections, because if you look at the ruminants, they ruminate, means they take and then they start ruminating. They go they just lie down in shade, and then they regurgitate, means whatever has been eaten is pushed into the mouth, and then they yes. So there is lot of bacteria. So these bacteria, they are responsible for the production of vitamin B complex. So we don't give vitamins, right? Whereas if your gut microbiome is good, then vitamin B complex would be there. And we know that for the nervous system, vitamins, they are the cofactors. All right. Vitamins are the cofactors in n number of process. Right. Next. Yeah. So, of course, we have completed this one also. Next. Go for the next one. Right. Now, so this is the gut and this is the brain. Coming to the cognitive dysfunction, what would happen? There would be increased apoptosis, increased mitochondrial dysfunction, increased hippocampal oxidative stress, and decreased hippocampal synaptic plasticity. I'll tell you what this synaptic plasticity is. Then and decreased number of dendritic spine, increased number of microglia, increased activity of microglia. Microglia is responsible for the fibrous tissue. Means wherever the neuron is lost, and then this glia, they would be taking the place of that neuron. It's something like liver fibrosis and renal fibrosis. So also you have neurofibrosis. So the increase in the number of microglia and increased activity of microglia means there are number of dead neurons, and that particular space is being occupied by the microglia, and also amyloid beta deposition, right? Mitochondrial dysfunction, yes. When I was a student, nucleus was the most prominent cell organelle. Now mitochondria is the prominent organelle. Able to follow that, right? Now, next is that no, not the slide. Right. Hippocampus is responsible for our learning and memory. There is a place in the brain, in the CNS, called hippocampus. There is a small part called hippocampus. It is responsible for our learning and memory, right? And now hippocampal synaptic plasticity, changes in the brain. I think you have seen a plastic chair, isn't it? Can you remodify it without applying heat? No, right? So also the changes that take place in the brain, our learning and memory, it results in plasticity, neuronal plasticity. That's what we say. And as I told you, you have dendrites. You have dendrites on the neurons, and on the dendrites you have spinous process. That's what we call, right? Whatever we learn and whatever we memorize is etched or it is written in the form of spinous process, right? Unfortunately, CNS cannot have this Parker and this Renault pen. So its way of remembering things is by making a spinous process, right? So that would not be there if the gut is affected. Means memory is being lost, learning ability is lost, right? Next. So the microbiome alteration or the therapeutic target through the diet, through probiotics, fecal microbiome transplants. We collect the we collect the fecal sample, and then in this fecal sample we try to isolate the particular organism, right? And then through the rectal route we inject into the colon of the patient. All right. Of course, it's a very common practice today. Because you being young, you might not have suffered, but those who suffer with this Clostridium difficile infection, fecal microbiome transplant is common. This one, it's almost like injection, right? Of course, it's not as pleasant as injection because it has to be injected intrarectally, and that too into the colon, right? Yeah. And you have supplements or medications, melatonin, curcumin, ginseng, berberine, and all that, statins, metformin, vitamin D, acupuncture also. Eubiotics, drugs which act as antibiotics or antimicrobials, they also help in favoring microbiome. So examples are nitrofurantoin, rifaximin, vancomycin, and other substances. Next. So this is a case of Parkinson's. Dopaminergic, if the gut is good, then increased dopaminergic neuron protection would be there, decreased pain or inflammation, decreased oxidative stress, and decreased behavioral changes, right? And in turn, brain influences by having good gastrointestinal motility, decreasing pathogenic bacteria and also decreasing gastrointestinal abnormalities. Next. Right. So these are the microbiome directed intervention. Just now we have seen this one: prebiotics, probiotics, symbiotics, postbiotics, Hodge therapy, and CRISPR-Cas9 based therapy. Next. Next, of course, we have come to this one out. Yeah, this is the last slide. Eat well, right? Eat well so that you learn well and memorize well. If you learn well, then retrieval is not a problem. If you don't learn well, then you struggle, but ultimately it does not come out. Of course, I do not know the frequency as to, but in our university, veterinary university, we for every 6 months, we have Viva OC, right? And then at that time, you should see how students behave, right? He behaves as if it's in his throat, but it's not coming out, right? Right? But that's how the pretension would be, right? Yeah. So, but if you learn well, you can memorize it well. Memory is nothing but retrieval. First of all, it has to be stored in proper shape so that it can be retrieved. For this, you need to eat well so that your microbiome would be good. Thank you.
Thank you so much.
I have this.
Thank you so much, sir, for your informative presentation on gut microbiome and neurodegenerative diseases. So if you have any queries, please feel free to ask.
Sorry.
Any questions from online audience?
At least you can ask me what to eat and what not.
Hello, sir.
Okay. Yeah.
Can we culture?
It was directed at the faculty just to stimulate our faculty in this particular research area, right? Microbiome is one of the upcoming research areas, right? Yeah. So, and for you, I had to just tone it down so that you'd not feel isolated from the lecture, right? Of course, right. Thank you, but thank you. Thank you for your this one. At least you were there in the audience. Of course, some of you, okay. Okay. Of course, it was a classroom scenario. I was feeling that I was in a classroom. So that's the reason why I went on and I encroached upon Vivek Tyagi's time.
Sir, I am really happy to hear this presentation. It's really informative, sir.
Thank you, sir. Now I request Dr. Vasudha Bakshi ma'am to please honor our chief guest, Dr. Kala Kumar sir.
But Tyagi would have come to Hyderabad.
Thank you, audience, for your time and presence here. Vivek Tyagi. Topic: the aims, rules, and functions and recent notifications of CCSEA for nominees and establishments. Dr. Vivek Tyagi has done his MSc in geology and PhD in entomology, presently working as a senior consultant in CCSEA, Department of Animal Husbandry and Dairying, Ministry of Fisheries and Animal Husbandry and Dairying, Government of India. His area of work is registration and renewal of establishments, breeding and trading of laboratory animals, and their institutional animal ethical committee related matters. Also processing the matters related to selection and training of CCSEA nominees. So let's welcome our Dr. Vivek Tyagi for the session.
Thank you, madam. Am I audible, madam?
Yes, sir, you're audible. Please continue.
Okay. You please allow me to share my screen first.
Yes, sir, you can share your screen.
Okay. I am really happy to be here as a representative of CPCSEA. First of all, I would like to congratulate the organization, especially the Anurag University, the convener, Dr. Vasudha Madam, Dean, co-convener, Dr. D. Krishna Prasad, and Dr. K. Sunan. And I am really happy that our senior member and senior nominee and the General Secretary of IPS, Dr. Kalakumar sir, is there as a chief guest. And it's really happy moment. And sir is there to guide us to be there as a chief guest. Now, I congratulate the organizers for organizing such a wonderful event on a hybrid mode so that more participants can attend this workshop. Now I'll start my presentation. Is it visible to all?
Yes, sir, it is visible.
It is about the CCSEA, its aims, rules, and functions of this body for the laboratory animal experimentations. Most of the people who are working in the laboratory animal experimentation field, they are aware about this CCSEA. And this lecture is very useful for the students and for the faculties who wish to start the experimentation on animals and to establish the animal house facilities. So we'll start what is CPCSEA and what is their guidelines and rules. Is it visible? My screen is visible and slides are moving. Slides are moving now.
Yes.
This is the first slide regarding the introduction of the committee. Okay. Madam, can you confirm that slides are moving or not? Now it's slide number 2.
The online participants, the slides are not moving. It is still in the slide number 1.
You please click on Dr. Vivek Tyagi presentation. 2 screens will be there on the computer. One is mine and one is the presentation sharing screen. Those who are not able to see my presentation, please pin that presentation. Is it working now? In the participants window, you can see 2 slides, 2 windows are there. One Dr. Vivek Tyagi and the other one Dr. Vivek Tyagi's presentation. You need to pin on the Dr. Vivek Tyagi's presentation.
Sir, actually, I'm already on the, yeah, now it's moving. Thank you.
Now it's coming?
Yeah.
Okay. So it is Committee for Control and Supervision of Experiments on Animals. In short, we spell it CCSEA. This committee is established under Section 15(1) of the Prevention of Cruelty to Animals Act 1960. This committee, CCSEA, is a statutory committee. Whenever we'll say statutory, it means this committee has some legal powers to regulate the experimentation on animals. Presently, there are 19 members in the CCSEA, wherein Dr. O.P. Chaudhary, who is the Joint Secretary of Animal Welfare, is the chairman of this committee, and the Joint Commissioner of Animal Welfare, Dr. S.K. Dattaji, is the member secretary of this committee. The aim of CCSEA is to prevent the infliction of unnecessary pain or suffering on laboratory animals. Now is it moving? The slide number 3.
Yes.
Yes, sir, it's moving.
Okay,
Sir,
perfect.
excuse me, sir.
Hanji.
Sir, can you please share this slide shares so that it will be clearly visible?
Means
Slide.
you want to show the presentation?
Yes, sir.
Okay.
Share the full screen, sir. Just click on the slide share.
I am showing full screen, but it's not working then. Now it's coming.
No, sir. Okay, but it's okay, sir. Can continue.
This is the problem whenever I am clicking on the full screen mode, na? Slideshow mode. Then it's not coming, the changing of the slides are not there then.
It's okay, sir. Please continue.
I'll make it an adjustment. So the duties and powers of the CCSE are given in the PCA Act 1960. Using these duties and powers, this committee has formulated one rule, that is breeding of experimentation of animals and control and supervision rules 1998, which has been further amended in 2001 and 2006 to regulate experimentations on animals. Everything what is required, the conditions for conducting experiments on animals, all things are there in these rules and their amendments. Under the provision of above rules, establishments which are engaged in biomedical research, breeding and trading of laboratory animals are required to get themselves registered with CCSEA and to constitute one committee, that is IAEC, Institutional Animal Ethics Committee. Mandate: the registration of establishments conducting experiments on animals, registration of establishments engaged in breeding of animals, and approval of animal house facilities for housing of small and large animals, permission for conducting experiments on large animals, conducting workshops, seminars, conference on laboratory animal welfare, and training program for the nominees of CCSE, who are the representatives of this body at the regional level, at the institutional level. These are the mandate of this committee. To fulfill its mandate, CCSE has also published some guidelines. The guidelines for laboratory animal house facility, how to construct the animal house facility, what are the requisite conditions, everything is there in these guidelines. Guidelines for constitution and reconstitution of Animal Ethics Committee, Institutional Animal Ethics Committee, IAC, it is also there. Care and management of equines used in the products of biologicals. SOP for IAC, how to operate this Institutional Animal Ethics Committee, it's also there. Guidelines on the regulation of scientific experiments on animals. A separate guideline is there for this. Guidelines for reuse and rehabilitation of animals post experimentation 2020. Guidelines for poultry and bird facilities 2020. Guidelines for experimentation on fishes 2021. And all the guidelines are available on the website of CCSEA. You can have a look of this website, it's also available. Is it visible, this website? These are the guideline section. Anybody from the online participants who have joined the conference through online mode can reply back. Okay. Like Dr. Anirudh is there. I can see so many names are there. Dr. Ashish.
Yeah, we can.
Yeah.
Yeah, we can see the website now.
Because otherwise it will be one-way bowling. I may not be able to understand that whether I am audible or not. Okay. Then these are the guideline section, and this is wonderful website, web page, everything, the requisite conditions, everything is there on the guidelines. Here you can see the Act, rules and guidelines section. There all the guidelines are there. Like compendium of CCSEA, everything is compiled in these guidelines. One small issue is there. Recently, the name of CPCSEA has been revised from the CPCSEA to CCSEA. Previously, it was Committee for the Purpose of Control and Supervision of Experiments on Animals. Now it is Committee for Control and Supervision of Experiments on Animals. Then other guidelines are also there. For saving of time, I am just skipping this information that you can download from the guideline section. Now, the breeding of experimentation on animals, control and supervision rules 1998. Rule 3A, registration of establishment for breeding of animals or trade of animals for the purpose of experiments. The institutions which are conducting breeding activities and trading activities of animals for the experimental purpose are required to be registered under Rule 3A. And some establishments which are conducting experiments on animals, however, they are not dealing any breeding activities or trading activities, they are just conducting experiments on animals, they are also required to get themselves registered under Rule 4A. The provision is there. And as I already informed to you that CCSEA, the statutory body, means has legal powers, then it is a legal requirement to get registered the animal house facility with this body. Then some documents are required for this registration. Application as per revised Form A is required to be submitted. However, the application is required to be submitted through digital mode through the website. For your information, I am just telling you the requisite details and performance. One blueprint and layout plan of the animal house facility of the establishment having the details of the various rooms with measurements for keeping animals, feed storage, washroom, office room, etc. as per CCA guidelines, and duly signed by the head of the institute and the architect with date. Here I have highlighted feeding storage room, washroom, office room, experimental facility. Everything should be specified in that blueprint, and that has to be approved by the head of the institute and the architect for verification purpose. Then minutes of the IAC meeting of 5 internal IAC members that they are recommending this animal house facility, any animal house facility for registration with CCSEA. Means this is their satisfactory recommendations. It means the internal IAC members, the members from the institutes are satisfied and they are responsible for the this recommendation of registration of animal house facility. Then the approval letter from the government bodies, like who are the parent or mother organizations of these animal house facilities. The birth and consent letters of 5 internal IAC members, again in the format is available on the web portal. The registration fee as per the purpose. Then it is clear that anybody and the establishments are required to register with CCSEA. Then under Rule 6, details of the experiments conducted is to be recorded. Every registered establishment shall maintain a register of particulars about the animal used from day to day for conducting experiments with the number of animals, the species, the age, gender, and other relevant particulars. It means it is also required to maintain the records. And for the record keeping, we have specific formats for the various type of records. Then stocking of animals. Under Rule 7, animal house shall be located in a quiet atmosphere, undisturbed by traffic, and the premises kept tidy, hygienic, and animals protected from drought and extreme of weathers. It's also required to be taken care at the time of establishing the animal house facility. Animal cages for small animals and stables for large animals shall be such that animals can live in a comfortable and overcrowding is avoided. In any case, overcrowding is avoided in the cages or in the stables for the large animals. These are the conditions which are mentioned in the breeding rules. It means if somebody is not housing the animals as per the requisite conditions, it is again illegal. Permission for conducting experiments on animals comes under Rule 8. The research projects on small animals. In CCSA, we have categorized the animals. The category of small animals means up to the level of phylogenetic scale of rodents and legomorpha comes under the category of small animals. And such projects are approved at the level of IAC, Institutional Animal Ethics Committee. And the large animal research protocols are recommended by the IAC and finally approved by CCSA in its meeting. One exemption is there, especially for the students. The research protocols dealing with withdrawal of or below. This is very, very important point. You kindly make a note of it. This is very, very useful point for the students who are working under the umbrella of such type of institutes. The research protocols dealing with withdrawal of or below 0.6 ml per kg body weight of blood from large animals on weekly basis. The frequency is also there, volume is also there, for the purpose of experimentation on feeding trials, breeding experiments, and livestock management studies, which require blood collection from the experimental animals to further study the clinical parameters, and for the purpose of clinical disease diagnostics, field studies, or thesis studies may be approved by the IAC also. It is the exemption which is for the institutes like ICMR, ICAR, veterinary and agriculture universities, and the government-funded institutions handling large animals. Such projects can be approved at the level of IAC. No need to send such protocols to the CCSE for the approval. Then it's clear that registration is required before conducting experiments on animals. The approvals are to be obtained from the IAC or CCSE as the condition may be. Then how to constitute this committee? It covers under Rule 13. For constitution of this committee, Institution Animal Ethics Committee, 5 members are required from the institute. And their designations are 1 biological scientist, 2 scientists from different biological discipline, 1 veterinarian involved in the care of animals, 1 scientist in charge of animal house facility of the institute concerned. These 5 members are required. Dear friends, these are the designations prescribed by the breeding rules. Anybody who is associate professor, assistant professor, or any designation can be designated under these designations. 1 chairperson of the committee and member secretary would be nominated by the establishment from among as the 5 IC members. Means, like you can nominate as a biological scientist, come chairman, or scientist from different biological discipline, come member secretary. One condition is there: if the establishment wants to propose its administrative head who is from non-scientific background, then the 6th IC members can be proposed. But that person, that administrative head of the institution, can play the role of administrative chairman only. Apart from that member, 5 names are required. In this condition, institute need to propose the IAC with the 6 members, where they for some different reasons. Sometimes the reasons are there for to apprise that senior head about the financial requirements of animal house facility, and so many institutes are there, they wish to appoint or propose that head of the institution as a member, as a chairperson of the committee. But if he or she is not having the background which is required for the IAC members, then it can be co-opted as a 6th member as a administrative chairman. Having a veterinarian in the IAC is mandatory for taking care and handling of laboratory animals. Nominees. Then after receiving the 5 names from the establishment side, CCSA nominates its representatives as a nominees of CCSA to these IAC of the establishments. Main nominee, 1 link nominee, 1 scientist from outside of the institute, 1 socially aware nominee. These nominees are required. The link nominee can attend in case main nominee conveys his unavailability in writing to the chairman of the IAC. Means link nominee is the substitute of main nominee. But one condition is there, please make a note of it, and this is very, very important point. Link nominee should be invited once in a calendar year in any of the meeting to apprise him or her about the activities of IAC. Otherwise, someday if you invite link nominee and he is not aware about the institute objective and aims of the IAC, he may not be able to evaluate the project. Therefore, it is mandatory to invite that link nominee once in a calendar year. And regarding that social event nominee, one confusion was there. Can anybody tell me the definition of the social event nominee? Anybody? Am I audible? Online participants?
Yes, sir.
How we can select the social event nominee? What is the definition? What is the criteria? Okay. The person from a different perspective is nominated as a social event nominee. The person who is having a different perspective in the IAC, whose concern is just looking after the welfare, the well-being of animals, not the merit of the research and all. So anybody who is having the different perspective towards the animals can be nominated as a social event nominee, and therefore a veterinarian can also be nominated as a social event nominee because they are having the better understanding of the animal biology and other things. So if we are nominating some nominees who belongs to the scientific background or working in the animal experimentation, sometimes they can also be nominated as a social event nominees. In addition to the above IC members, a specialist may be co-opted while reviewing special projects using hazardous agents such as radioactive substance and deadly microorganisms. But that co-opted members will not be the part of decision-making team. He or she will just offer their advice, and that advice will be recorded in the minutes, and minutes will be signed by the approved IAC members only. Then it's clear registration is required, constitution one committee is required, and how to constitute this committee, it is also conveyed. Then what are the objectives of this committee? What to do after constitution of the committee? First and foremost objective of this committee, that experiments to be avoided whenever it is possible to do so. It is a responsibility of IAC. Then experiments on larger animals avoided when same results can be achieved in smaller animals. This point is also to be adhered at the time of scrutinizing the research projects. Then most important thing, rationalize uses of animals like 3 R's. First of all, we should see the replacement of animals. using non-animal method or any scientific methods employing for non-sentient materials which may replace use of living vertebrates. That can be seen whether the possibilities of replacement is there or not. I have seen the schedule. Dr. Muhammad Idris is there. You can utilize the expertise of Dr. Muhammad Idris sir for this purpose. Reduction. If this replacement is not possible, then we should see the reduction in the number of animals which are used to be obtain information for given amount and precision. But justification is there. Justification should be there. Like you cannot just offer vague comment that you reduce the n number from reducing two animals per group. You should say why you are recommending that reduction with a statistical background, with statistical justification. Then you can offer the reduction in the number of animals in the project. Then if it is reduction is also not possible, then the researcher should see the refinement of experimentation design methods to reduce pain and distress to the animals. Then again, the important point, animals to be properly looked after before and after experiments. It is also the responsibility of IC to check the fate of the animals. Suitable records to be maintained. This is also the responsibility of the IC. Thank you for this presentation, for hearing this presentation. If you have any doubt and question that I am happy to replying it.
Good afternoon, sir.
Yeah, good afternoon, madam.
Myself, Jabin Fatima, I'm research scholar from Anurag University.
Ji, please, madam.
Actually, my research area is Alzheimer's disease. So I was planning for ICV model for inducing through the amyloid beta, but have unavailability of this stereotaxic apparatus. So I was planning to work with a manual model. What do you suggest? Because I've gone through various procedural videos demonstrating the manual method also. So is it possible that I can work without the stereotaxic apparatus?
You can work and this can be discussed with your IC.
Okay.
If IC permit, then you can go ahead.
Okay. Actually, I've come across 2-3 videos and also live demonstration also. They are showing it the bregma just 2 mm down. They have shown very clearly how to locate the point and drill. So I was thinking, is it possible or not?
Dr. Kalakumar sir is there. Sir is there or not? Sir is there, na?
No, he left, sir.
Okay.
Okay, sir. Thank you.
Thank you, madam.
Any questions from online audience?
No, ma'am.
Okay, thank you, sir. Thank you, Dr. Vivek Tyagi, sir.
Thank you, madam. Can I leave the meeting, madam?
Yes, sir, you can leave. Thank you, sir, for your time.
If anybody is having any question apart from this presentation, they can also write an email to CCSEA, and I'll be happy to reply it.
Okay, thank you, sir.
Okay.
So with this, we have come to an end of the day 1 of FDP program. So please fill the feedback which will be provided to you through the WhatsApp link. And please join us tomorrow at sharp 9 a.m. And thank you.
Ma'am, hello.
Yes, sir.
Ma'am, I'm an online audience, ma'am. I think my name is not there in the WhatsApp group. It has not been added, ma'am. Can you just post the feedback link here itself? If you don't
Yeah,
mind,
sure.
or else.
Sir,
Yes, ma'am.
sir, can you please communicate any of the faculty members or our co-conveners?
Yes, ma'am. I will. I think Dr. Krishna sir is there in touch with me. Can I send him? Can I speak to
Yes,
him?
sir. You can send your number to the sir. We will add you in the WhatsApp group.
Okay, ma'am. Right. Thank you.
Thank you, sir.
Thank you.